I'm Helen Moore. I'm a mathematician, differential geometry by training, but and I got PhD was 28 years ago, and a lot has happened in the meantime. A few years after my PhD, I switched to doing applied mathematics, and so I'm going to talk to you about some of my work today. And also, I should say, I spent the first 11 years after my PhD in academia, and then I moved to the biopharma industry and spent 15 years there before returning to academia in 2021 and joining the Laboratory for Systems Medicine, which sits in the College of Medicine at the University of Florida. So I think you all know about the group. So my perspective is colored by my time and experiences in industry. And I will also just mention that most of my work that I did in biopharma, I just couldn't publish most of it. So there's that. But what I'm going to talk about today is some perspective on systems models and how do we evaluate those. And the very large number of the models being used in the drug development work, which is being done in industry, but also in academia, some at University of Florida, and also the regulatory agencies like the Food and Drug Administration in the US, so the FDA. They're all using models. Models have been used for, figuring out all kinds of things, streamlining clinical trials, to determining when to give Paxlovid. So all of these are model applications. And of course, so I'm going to be very broad in my my talking today, but I'm going to also say that much of what I'm going to present is written down in a manuscript that my collaborators, Sitze Brackman and Pras Pathmanathan and I wrote. And I'll just say the origin story of this is Sitze and I were working together on some projects when I was at a company, and he was at MathWorks at the time, helping with some MATLAB and symbiology applications. But I had this idea, and so we started working together on a kind of analysis and a manuscript, and it completely changed directions, so like 180 degrees from what I had originally proposed to him. And in the meantime, we also ran a session at a conference, American Conference on Pharmacometrics, where a lot of this work is highlighted often. It's held every fall in the US. And we recruited Pras Pathmanathan as one of the speakers. And we also recruited Bill Oberkamp as one of the speakers. And he's one of the authors of this very large tome on verification and validation in modeling. So there were our speakers, and then we got Pras to come in on us and write some sections for this manuscript. It became a very large piece of work, and so that's published now. But so I want to acknowledge my co-authors there. And Sitze is now at AbbVie, and Pras is still at FDA. And I also want to then share some of my motivation for this work. So my other research areas outside of model evaluation include predicting response to immuno-oncology therapies. So this is the one paper I got out of 15 years of very intense work, lots of projects, very fast. This was work that I did at AstraZeneca, and I had developed a method for, when I was at another company, I developed a method for predicting who's going to be a responder to immunotherapy in the cancer space. And this model-based prediction gave very good results. So the error rate's around 10%, looking at an early time point at 12 weeks. We could tell in advance who was going to be a responder and who was going to be a non-responder. And so that's published. So that was the one piece of work I got to publish. And then this other work that I really is my major focus is optimizing combination drug regimens. And just one quick example for the case of HIV therapies, there's here you can see two different regimens. One is traditional, I'm not showing the regimens, traditional constant level dosing. The other one's optimized, same total amount of drug for the patient, different schedule. And we can compare the outcomes. In this case, the black curve is the good guys. These are the CD4 positive T cells, and they're healthy. And then the purple is the bad guys. They're controlled in both cases, but there's a significant difference in the outcomes for the good guys, these cells, keeping them out of this dangerous regime for the patient and ending up with about 70% higher value So just with a change in schedule, we can achieve that. And that's not the best that we can do. We can do even better. So in all of this model-based work, we were developing models with the aim of impacting outcomes. So we want to give a prediction. We're going to use that. We're going to have some actionable prediction. A lot of the questions came about, well, how do we know we should use your prediction? Okay, well, it's a fair question, and I care about that too. So how good are the models? How, can you evaluate those models? So that really motivated this work on the evaluation. And I'm going to also explain, because I use this terminology and in the paper we use it, I'm going to talk about a quantitative systems pharmacology model. And the quantitative piece refers to it being mathematical. So in our world, a mathematical model has at least one equation. Of any sort. And then a system is something that incorporates some level of mechanism. It can be very semi-mechanistic, but it could be very, very mechanistic. And then we say that the pharmacology piece is where we are using that model. It doesn't necessarily have a therapeutic effect in the model, but it allows the exploration of that. So it might just be a disease model, and you've got a lot of pathways, and you could explore those pathways. So that's the QSP, Quantitative Systems Pharmacology model paradigm. And also to share with this group, there's some kind of clear results of the cost-benefit potential for these models. There's some estimates. They are updated at various times, but in the US, it can take over a decade, more like 15 years, and it can cost probably now about $2 billion or more to develop a single successful approved drug in the US. So that has to be safe, first of all, to get the approval. And then it also has to improve efficacy for patients. So given whatever's out there already, does this make any difference? And so after all of those thousands, tens of thousands of compounds that are considered that don't work out and that might even get to the point of a phase 3 clinical trial, the total cost to get one that ends up being successful is about $2 billion, $1 to $2 billion. And so if we use modeling, maybe we could save some of that time and resource. And in fact, it's very clear we can. Some very early examples show this. Here's one example. Use of a QSP model saved about 40% of the clinical trial time and 66% of the cost for a phase 1 trial. And this was an example of a type 2 diabetes drug and new mechanism. So the modeling was very clear, very, very clear. The modeling was saved them from having 4 of the arms in the trial and shortened so it shortened that time for enrollment and also the cost. So, and then also we know not only can they save time and money and other resources like patients who are sick being experimented on, but they also can result in better outcomes. So we can get better efficacy, we can get better safety by using these types of mechanistic models. And the FDA is also on board with this, and they have various publications. Here's one quote, that these published QSP models have demonstrated the utility of this modeling in pharmaceutical research and development. So, so now QSP models have a range of uses. So we use them to find drug targets, combinations of pathways to target, and we would estimate the uncertainty in a predicted outcome. So if you're designing a clinical trial, you can use that, a mechanistic model to make predictions about settings we haven't seen yet. And all kinds of decisions like around that. And they can also determine data that we need in order to reduce that uncertainty. If we make that estimate and decide that's not something that we can move forward with, so we go back and we run some more experiments, reduce the uncertainty, and now we can say, okay, here's the right path. So a lot of these uses and others can be supported using sensitivity analysis and uncertainty quantification. So that's kind of the theme of this presentation. So now there are also, just to explore why QSP models are not used more, there was a study done. And well, first I'll say that QSP models clearly can improve drug development. So they can get faster and better results for that. So that means they're being produced. So here's my little QSP, my mechanistic model for QSP models. And there was a survey a few years ago, and in this field, this was a large sample of the field, over 100 QSP modelers. And there were 3 major reasons why they're not being used more. And one was not enough modelers. I'm not really going to talk about that today, but that's one thing. Another was there's budget and infrastructure limits. And then the third was the lack of management interest and support for the models. Now, I actually will, so those are the 3-sided models, but this was a non-exclusivity choice. So you could choose multiple reasons. And so I actually think that this, the lack of management interest and support is the cause of the decisions that lead to budget and infrastructure limits for not supporting more modeling there. So I'm going to just limit it to this. And then if we think about, well, what's causing this lack of management interest and support, it's really, I would say, a lack of confidence in QSP models. So that goes back to saying that, this, so that reason I'm not going to address, but this reason is really an important one for today. And I'm going to posit that it's the standards for evaluation of these models that would lead to that the lack of standards is blocking our confidence in these QSP models. Okay, so that's what I'm going to focus on, the standards today. So methods for model evaluation. And here's a figure from the paper that was sent out, the link that was sent out for this basis for this talk. And let me just walk through a little bit of this. And so in this diagram, this top level is talking about inputs. So all the gray are inputs or outputs in this schema. And these inputs at the very beginning, the aims, scope, and risk, this is really the context of use. So we're setting the context of use. What's the goal of the work? And then to what extent are we going to dive into that modeling? And how much time, so scope is how much time do we have, how many modelers, or if you have one modeler or more, what is the expertise level of the modelers, what kind of access to computational resources. So all of that is the scope. And then the risk is, how much are we going to rely on this model for the outcomes versus use it in conjunction with other evidence. And maybe it's just going to be a supportive piece of evidence, or maybe it's going to be foundational. We really need this to be very, we need to have a lot of confidence in the model because we need to use it for the predictions. So all of that gets decided at the beginning, and then we make a strategy. So the goal would be something like a question we're asking, and then we figure out what are we going to do to actually answer that. So we'll have the scope here, this outline of the model. We will think about how we're going to do our model building. So we're planning that, and we do that work. So here's the project planning, model building, and then and the planning also includes the analysis and evaluation. That's here in this section. The model building is going to give us the initial model and parameter distributions. So we come up with that somehow. Now we may use some initial data to do that. So there's probably a couple more places we could put this. We just didn't want to make it too complex in the diagram, but the data, any data that we have that we could use, we could start with to determine distributions, nominal values, some information about the parameters using Bayesian inference to get those parameter values in the models. And I'll say that, in the QSP world, the vast majority of these models are systems of ODEs. So sometimes I might say that, but in general, these principles are applying for all kinds of models. So we initialize, and so we get that model and parameter, and then these steps, these really important steps. In fact, I would say that Bill Oberkamp really, really emphasizes the verification a lot, in addition to the validation. But in our world, in the world of ODEs, we don't worry as much about the solvers. So the verification is really restricted to, does the code reflect what we planned when we drew that model diagram, when we all came to an agreement as a team? Is that coded correctly? We do some unit tests, we do some system tests, we do all of this testing. So that's our model verification. Once we've done that, we have our verified model. Then we can start the real exploration of the structure of the model. So we would run sensitivity analyses of various types. That's going to give us, at a high level, it gives us out of our list, I'll give an example today with 35 parameters. So out of that list of 35 parameters, it might identify, for example, 8 of them that are the most influential parameters. So, so that's the output of our sensitivity analysis. And then out of that short list of 8, we go and we see, well, okay, that's great. That's very nice that those are the most influential parameters, but can we actually estimate them? So we do identifiability analysis of various types, and we determine whether or not we can estimate them. And we might have to shorten our list. Instead of 8, it might be 5 that we can estimate. And then we go forward and we will use our calibration data to estimate those 5. And we can have some confidence in those parameter estimates because we've done our homework, we've done our due diligence, and we have confidence that those are important, they're influential, and we can estimate them. So we do that estimation, we calibrate the model, and then we look at we get those parameter estimates with distributions, and then we can perform uncertainty quantification. So we have our distributions, we just basically run it forward and see what are the outputs of interest and what are the ranges that we get there in terms of the possible values. And we have distributions for those too. So that's our uncertainty quantification. The gold standard for the validation step is to have a comparison with data. So we have ideally external evaluation data. If we're not lucky, we don't have enough data, we might reuse our calibration data. So we'll do cross-validation or hold out some kind of structure there. But we would need to plan that in advance to avoid the bias of, oh, now that we've seen the model and seen what it can do and what we think it's going to do, now we decide which data we should use. No, we should really make that decision up front. FDA is very aware of these types of biases. It can confound the results. So then we get our outputs, we've got our final model and the parameter distributions. And I will say that I'll point out here that at any point we may need to iterate. So for example, we may have too many parameters, and we can't identify enough of them. So we might go back and reduce our model. So we have all kinds of steps we can take to work around this. And once we do, we are satisfied. We have our final model. We're going to document it. I like to say that another step of validation is publication. So when you put it out there and you have reviewers look at it, then I think that's a really important step in the validation of the model. Okay, so, and I'm going to go more, I guess, yeah, let me just go ahead and move into the 2 topics I'm going to focus on. So those are sensitivity analysis and identifiability analysis. So I'll just put this table up here from the paper, so you can look that up. I'm not going to go through it, but we do talk about the different methods, local and global, and the pros and cons of the different methods, and some of the computational estimates for those. And so there's a long list of methods. Some of the ideas that we go over are, this local versus global, local being done at a single point in the parameter space. So that's giving us information locally. Global would mean we're getting information across the parameter space. Sampling could be done one at a time. You change one parameter at a time to calculate that sensitivity index. And then we could do the all at a time, which is where we are we're taking points in the parameter space, not just moving from one point to in one parameter, changing one parameter or another. We're actually moving multiple parameters at the same time. We're moving through the parameter space where things are changing. So we're not holding all of the other parameters fixed while we move one of the parameters or more. And so there are different methods to do this. And we also have the variance-based methods or derivative-based. And I just want to make one point, perhaps, really clearly, the local versus global. So for example, so I want to kind of make a plug for global sensitivity analysis. And here's a particular model in our field, not necessarily so mechanistic, it's semi-mechanistic. So we've got a 1-compartment model, you've got oral dosing, and there's nonlinear clearance. And when we do a local sensitivity analysis, we can pick one parameterization. So this is a very toy model. We've got parameters in the model that we've set equal to 1. Here's the 4 different parameters that are in this model, and one of them is set to 10. And so that's the one here. And so we get one type of result about the influence of the different parameters, and higher is more influential. So here we are saying that this parameter VD in the model is the most influential, and then these other here are not as influential. KA is the least influential. We do a different local sensitivity analysis, and we find out that a different parameter is determined to be the most influential. And so the rank order of these influential parameters is different, very, very different. So the question is, which one do we think is right? And actually, this speaks to the sense that we should use global sensitivity analysis, so when would we use local sensitivity analysis? If our model is linear, then we can use local sensitivity analysis and be confident that we're getting information about even if we're wrong about the parameter estimate. If our model is additive, we can do the same thing. We can separate the parameters in that way. So that would be a valid time to use local sensitivity analysis. Or if we have very precise and accurate parameter estimates for all parameters, that would be another case in which local sensitivity analysis would be adequate. If we're not in those settings, we would want to use local sensitivity analysis as a first step to moving to global sensitivity analysis, especially with these large models. So we're going to try to rule out some of the parameters as not being influential. That we can do with this. And so when people ask me, well, when would you use local sensitivity analysis and leave it at that? Like that's what you use in your publication. I would say, well, I would use local sensitivity analysis if I didn't have enough money to buy a computer. That's when I would use it. Otherwise, for these nonlinear, for these biological systems, it's not typically what we recommend unless it's just the first step, but it's not the one that you use in your publication to say, hey, look, I have done sensitivity analysis on my model. I've determined the most influential parameters. Okay, so that's my take on global sensitivity analysis or sensitivity analysis. And let me just talk a little bit about identifiability. And here I've got another toy example. So I've got a linear system, linear algebraic system. And so what I've done here is I've pulled out this matrix. So you can see the system there. I've made a very simple one. I used Desmos to make this. And so you can see it's 2 lines. And when we think about identifiability, the way I want you to imagine this is related to our models, our biological models, is this is when we've collected some data. So we've got, so we have some time points, we've got some values in some of these things, and we're trying to estimate the parameters. The X and Y here should represent unknown parameters. Other things should be known at this point. And so, so then we want to know, with the data that we have, with the information that we have, can we identify these parameters? Can we actually find unique values for these parameters, the X and the Y? Well, in this linear system, linear algebraic system, all we have to do is take that associated matrix and we compute the determinant. Here it's -5. So the fact that it's not 0 means that we can identify X and Y. So, and in the graphical system, what it means is that the determinant not being 0 means that these lines intersect. And so when we try to find X and Y values, we can uniquely find them, the ones that satisfy the system of equations. Okay, so if the determinant is 0, then either the lines are parallel or they're overlapping. And so in either case, there's no unique value, and sometimes there's not even, it's not even possible to find values. But in a lot of cases, in most of our systems, we have underdetermined systems, so we've got lots of possible values, just like if we have overlapping straight lines. Okay, so identifiability, I want to say I have seen this in my field. I have seen this. Identifiability, I will not say it's identifiable if we say, oh, my fit converged. I got parameter estimates. Nope, that's not identifiability, despite what some people have said. But so we also think about 2 different ways to look at identifiability. There's structural, which is the first test. Is it even potentially possible to estimate the parameters? And if we can't do that one, we just go home. We're done. Okay. It's not even possible. But if we do pass that test, then we move on to the practical identifiability. What about with the available data? So So of course there's some caveats here, but so we think about identifiability. Is it possible to estimate these parameters? And so we've focused in the example I'm going to show, which is another paper, we focused on structural identifiability first. And so we looked at a few different methods. There's combos, there's DAISY, there's GenSSI. I could say a few words about these if people are interested, but they're different methods and they're different strengths and weaknesses of them. We tried all three in the model I'm going to show, in the example I'm going to show you. And we ended up.
Sorry, could you possibly define structural identifiability? Because that term sometimes means different things in different subfields.
Okay, so let me go back and say, if we have a linear algebraic system, it is simply a determinant calculation. We have identifiability that structurally it's possible if the determinant is not zero.
Okay, so you're not talking about hypothesis testing for model structure. You're talking about is it technically possible to identify parameters within a given model structure?
That's exactly
right. To be very clear, because it's just that different fields use that term slightly different. Thank you.
Oh, that's good to know. I really appreciate you letting me know that. And thanks for the question. So yeah, so that's what I'm just going to be strict, very, very minimal requirements.
And for your next slide, the one that you had after this,
yeah,
seems to suggest that what is the caveat for parallel pathways?
Oh, yes. So we could not identify parameters if we have multiple ways to get to the same thing and we're only measuring output.
Right. That's what I thought. Okay. Thank you.
Yeah, thanks for that question. I appreciate that. I hadn't thought to say that more. Yeah, so these are different methods. There are, polynomial, nonlinear multivariate polynomials with the Grosvenor bases are being used. And this, the DAISY is differential algebra, kind of similar. And then the generating series, it's using Lie derivatives to kind of it's using the fact that any function, any analytic function can be expressed as a Taylor series. And so what we do is we can then differentiate it. So we can get lots and lots of equations. Think about our linear system before. We get lots and lots of equations by taking derivatives on both sides. And then we can get relationships between parameters. The more equations we have, the more relationships between parameters. And some terms may drop out. So we get better and better, closer and closer to being identifiable. If there's an equation that just has one parameter in it, then we're done. We know that parameter is identifiable, but we take lots and lots of derivatives. Oh, I'll show a picture of that. But okay, so there are different methods for doing this. And I should say with the GenSSI, it uses Lie derivatives, so it finds the directions of the that are advantageous for computing the derivatives. So it finds the easiest directions for the derivatives. So the computations are very feasible. So that's the identifiability. Let me talk about my example here. So very, very simple QSP model, semi-mechanistic, and it's a model of multiple myeloma. M is the representation of myeloma cells. It's actually M protein levels that is correlated with the myeloma cell disease burden. And then we've got natural killer cells, we've got cytotoxic T lymphocytes, and we've got regulatory T cells in the model. We've got lots of arrows for influence, and the equations reflect that. But for example, okay, let's see, A for example, would be IL-2, and B is represented from the interferon gamma that the NK cells produce, and they both increase proliferation. So there's crosstalk between cells. So that's the kind of level of mechanism that's in this model. And what we're trying to do is determine some of the key drivers in the system. So in fact, sensitivity analysis is used not only to get more confidence that the model is doing what we expect it to do, but also to determine where we should go after these pathways therapeutically. So we did in this paper, yeah, I guess the model development is in one of the papers. So there's lots about how we develop that model and some of the analysis, like dynamical systems analysis and equilibrium and stability there. And then in this paper, we did the sensitivity analysis, and we use two different methods. This is just the sensitivity index on the left, and this is the partial rank correlation coefficient on the right. And we've rank ordered them according to their level of influence. But so then we mapped it for all 35 parameters we looked, and one of the really striking results was that when we were looking at the top 10, which was our plan, we noticed that 8 of them were the same, appeared in both lists, I should say. And so I'm showing you by having these little line segments to show where they are in each of the lists. And so what we used was this kind of consensus method to decide. And that was also part of our plan, to use a consensus method to decide which parameters we would consider the most influential. And so we took those, and here's just a heat map of where they appear in the model. So we have the red being the most influential, orange being second most, yellow third most, and so on. We've got the 8 most here in this heat map. And then if we really want to think about the application, the therapies, and here's just 6 therapies out of about a couple of dozen therapies that are currently approved for treatment of patients with multiple myeloma. And with our collaborators, we've got clinical people and experimental people in here. We developed this model for where the therapeutic interventions occur. And so, for example, elotuzumab is, it's got a particular mechanism. It inhibits, it's a SLAM F7 inhibitor. And so it affects the efficacy of the NK cells in their blocking, in their increasing the loss of the multiple myeloma cells. I guess it's easier to indicate here. So that's just like, this is a loss. If we could increase that loss, that would help us. So that's one of the pathways that's important in the top 8. The loss itself, if we just could have a therapy that went directly for the loss, that would be the best. So we've got a few of those in this list that can do that. Bortezomib here, down here, protease inhibitor, and then these thalidomide analog and the dexamethasone, kind of the general immune suppressing therapies. So we've got all of that. So we can then make our choice. And in fact, should say that this work was done in order to do some optimization. So we couldn't do the optimization for 25 therapies. So we had to subset them. And we used the sensitivity analysis in order to subset them. And then we could do the optimal control and optimize the therapies. But then here's the sensitivity analysis. There's a lot of information there, in addition to having our clinical collaborators corroborate that, okay, some of these they thought were very important. Of course, we know if you can increase this loss of the myeloma cells, that's great. But what they hadn't realized was if you can keep the number of NK cells high, that would be a really important feature. Same for the cytotoxic T lymphocytes. So you don't have to decrease the number of Tregs. That's not as important as increasing the anti-myeloma immune cell types. Okay, so
Helen, I do have a minor question here. So
to
me, just from the biological perspective, right? If the killing is important, then the proliferation, the production, because their symmetry supposedly to be important. If you stop their proliferation completely, the parameter 5 here, right? The process 5, where it's the tumor cells reproduce themselves. If you can stop that, then I naively would think that can be very important for the tumor, but it seems not shown up in the sensitive analysis. So I'm just a little bit confused here.
Yeah. Okay. So let me explain a little bit, because that's a very good point. I haven't said anything about that. So in fact, what we think of is this source term not depending on what the immune cells are doing or the therapies, because we think of it as in a protected niche. So it might be what we would consider a stem cell population. And so, yes, we could slow down proliferation, but killing is really, really important, because, if you're having this unstopped production, you need to always kill. So it's like, for example, there's a disease that has a therapy that does this, or actually it blocks proliferation. So in chronic myeloid leukemia, there's a therapy, imatinib, that blocks production of, the general production, not the stem cells, but the constitutive unchecked extra production of these leukemic cells. And so that works, but only as long as you're not resistant to that therapy. And then once you get resistance, then, okay, that's not going to work anymore. So, but that's really essentially playing the role here. And so that would work. That would keep it under check. But we also would need some killing in this disease. This disease here, we need killing. And the other disease, we don't necessarily. So it's different aspects. Like if we, for example, if we could do lots of killing, but it wasn't maybe the fastest rate, that production could be fast enough that the numbers get large and still cause symptoms, and mortality could still be bad. So in fact, I would say that from these systems, from the diagrams, I would say it's the nonlinearities and it's the size of the effects and the relative size of effects that make it not possible to have an intuition. I mean, maybe the intuition is, yeah, we should kill lots of myeloma cells, but like, what are the other things that are important? I wouldn't have known that. I mean, the Tregs are very important in this disease. There's also some other cells, and we're kind of lumping their effects together in this one cell, representative cell type. And so I would not have guessed that it's not so important. It's not in the top 8. I would have guessed it was in the top 8 to do something about the Tregs, but it turns out in this system, it's not, according to our model.
And if I could make a comment about like scaling up, which I think you're probably going to get to, right? Because there may be circumstances in which the killing is not the optimal strategy, just as you mentioned, right? Given the combination of toxicity, delivery, how much you can actually affect that, right? So a global optimization for like the total system, whether or not that system survives, incorporating all the non-tumor processes that are present, you would have to optimize over that particular function, because that's actually what you care about, right?
That is exactly right. Yeah. And so that's a really great point. Thank you so much for bringing that up. So this sensitivity analysis is done with the goal, the endpoint of efficacy. We want to reduce M as much as possible. But the optimization over here on the other side, when we take those therapies and we do an optimization, we actually have terms that account for efficacy, but also terms that account for toxicity. And so that's exactly right. This on the left does not reflect the toxicity. So yes, it may be best to do this killing, but there's going to be the collateral damage. If we kill lots of cells that divide rapidly, like general chemo does, we have big hits. And so we're limited in the dose we can give. That's a great point. Thank you.
Right. And also the mechanisms of a resistance development to whatever it is you happen to do, right?
That's
because you might have more evolutionary escape from something that's designed to kill you as opposed to suppressing proliferation.
Exactly. Yeah. Thanks for all of that. And so that's incorporated over on the right-hand side. Yeah. So not on the left. Really, really good points. So we could think about an outcome where we do a sensitivity that kind of balances those. But a lot of times we think about in our system, like, what are the new drug targets? If we just think about what we have already, and some of them are chemos, and so they're going to have the side effects, then we're kind of limited. But if we in this analysis, we're a bit idealistic, because we say, what if we could get a drug that targeted specifically myeloma cells and only killed myeloma cells? And we have therapies like that are getting closer and closer to that. So that's why we do the sensitivity analysis in that way, because we're often interested in what about future therapies too. Very nice. Okay. So then we did, for these 8 parameters that are the most influential in this model, we also did the identifiability. So out of those, is it even going to be potentially possible, just from the structure of that model, like where we have data that we're going to measure and where we have the parameters, will we be able to estimate those 8 parameters? So this is, the structural is, if we have complete time series data, can we uniquely identify those parameters? And so it turns out the answer is yes for those 8. And anytime we added one more on those lists of the most influential parameters, we got an answer that says it's definitely not uniquely identifiable. It's only locally identifiable. And so this is just the representation from GenSSI, and it represents a little of what I was saying for the mathematicians. So each row is an equation, and you see the 8 parameters here. So each column is a parameter. So the black means we've actually got that parameter in this equation. So there's a non-zero coefficient for that parameter. And so here's an equation that has 4 parameters. Here's another equation that has 2 parameters, just these 2, the RC and delta C. And here's another one that has RN and delta N. And so we keep taking derivatives. And if we get enough equations that we can uniquely identify those parameters, then we can stop, and we know we can uniquely identify them. The problem with this method, although it is probably the most robust of the ones we tested, is that we don't know in advance how many derivatives you will need to take to determine whether or not it's uniquely identifiable. So you might have set a certain number, and it's not within that, but it says it's not finished. And so then you have to run it again with more, and it's very, very computationally time consuming. And the other methods, some of them are quicker, but again, they can't work unless you have rational functions for everything, for one of them. And the other is a web app, and it was quite limited in what it could do. So it seems like a good method, but just guess a big number and let it run for a long time. So structurally, we do have identifiability of these 8 parameters. So that's the good news. And then we move on to practical identifiability. The bad news is, we were not able to get the clinical data that we had planned, and it had to do with who was at the company at the time. So things changed in terms of data availability. So this practical identifiability reflects only using literature data. And as you can see, so we have very, very little literature data, and we didn't have any time course data. So we had 4 steady state values, and it was the 4 cell compartments I showed you previously. And so with those steady state values, we use this Markov chain Monte Carlo, and actually we're using Metropolis Hastings for our deciding if we're going to accept or reject based on what we expect for how close they are to the actual data we have, the distributions of the data that we did have access to. And so you can see there's no practical identifiability. If there were identifiability, we would have vertical or horizontal lines for any of those parameters. And we don't, but we do have some relationship between 2 parameters. So the delta M and RM are in this with the other parameters frozen. In this setting with this data, we have some relationship there. So that's pinned down. So that eliminates one of the parameters. We can just reduce it to 7 parameters, but we still can't get everything. But there are some relationships, as I said. Okay, so this is not only are we learning about the data and making sure at each step that this matches the data, matches the qualitative behavior that our clinical collaborators can tell us. We've got, on this paper, we had, I think, 15 people. And so half of those are people from the clinical, and then we've got experimental people. We had a medical director, program director, clinical pharmacometrician, an actual pharmacometrician who's doing models, pharmacometrics models, all of that. So they, as a group, as a team, they were able to say, from this information we're getting from the sensitivity analysis and identifiability, they're saying, yes, this matches what we see in the clinic or in the experimental settings. So to kind of wrap up, I would say that the standards for the model evaluation, they can result in better models. So we had some iterations. I didn't show that, but we have iterations, and that's typical because we get a sensitivity analysis and we realize, oh, there's something that's not showing up. And so should we include some other feature in the model? Let's go back. Okay, now it shows up. Great, we're going to include that feature. It's an important one. So it can result in better models, but also these standards having a set of analyses that we will run for each model, or having a plan for the particular project and the model we're going to use for that project, that can help with the model evaluation by stakeholders, people who are not modelers. So when we can show results and say, here are the most influential, here they are identifiable, here's the estimates from the parameters, we have confidence, here's the uncertainty quantification, which I didn't show, then we can say to the stakeholders, here's the evidence we are using to make a decision. And so here's the evidence, you make the decision. Is this good enough for the purposes of this model? And in particular, when we say stakeholders and timely model evaluation, we're thinking of FDA. FDA gets these submissions and they have 30 days to turn around. They have all the data package. It's enormous. They get all the modeling, models, code, and results, lots of figures. And so if we have a standard set for evaluating models, that would be helpful because, and then they can just look at the results. And we have those for some of these other types of modeling efforts, statistical modeling. We have a lot of diagnostics that are accepted in the community. So we have a standard for model evaluation for those. And so that would help with these other mechanistic models. And so if the stakeholders are more excited about it, then that's going to increase this use, and that will reduce the time and cost and improve the outcomes. And it will also, my ulterior motive, as I stated at the beginning, I wanted to use models, mechanistic models, to make predictions of other things. So I want that patient response. Who's going to respond to an immunotherapy? Hard problem. So let's use some models to try to help with that. How do we come up with regimens that are optimal? Hard problem because it's combinatorially just too hard to do the experiments in mice. And just to summarize the things I was talking about, so these standards that we propose in this paper, we've got sensitivity analysis, we've got identifiability analysis, we've got uncertainty quantification, then of course the validation step, comparison to data, and another that I would claim is very important for validation, which is peer-reviewed publication. So that's what I wanted to share with you. I'll leave it on a references page for all of the things that I happen to mention. And I'll probably have to put that other model on there. But that's it for what I wanted to present, and I'll take questions, more questions.
This is great. Thank you, Helen. My apologies for being late. We have some time for questions.
Great talk. Very,
Thank
very, very interesting. So when I got introduced to QSP, it sounded very similar to the sort of things that I was interested in, but I'm almost, and this, so clearly this is an extraordinarily beneficial set of tools and approaches, but whenever I meet someone who does QSP, right, there's initial supposition that the thing kind of works, right? Or with your representation of the system, that there is a supposition that you can actually find something that will work in the overall context. And, I correct me if I'm wrong, but it doesn't seem that that's a primary focus of what QSP does, because most of the actual, work that I've seen in it either assumes a particular, generative mechanistic causal structure, right? And then operates within that particular structure to say, given this structure, if I do X, I would expect this particular outcome, and you validate that, experimentally or whatever. And then subsequent applications are related to the things that you talked about with respect to, local and global optimization. Is that a fair characterization of kind of what mostly this involves, or am I restricting it too much?
So let me paraphrase to make sure I'm getting what you wanted to ask about. So you're asking if the models, because we are making mechanistic models manually, if we might miss some hypotheses, and so we're not testing for those, and there might be some confounding factors, and so we might think we've got a model that captures the mechanisms, but we don't.
Right. That's right. Because this, I would imagine, is a very common complaint against all these models, because they represent a choice, a selection within this cloud of epistemic uncertainty as to, I'm going to make a model assuming this is how I think it works, right? And we always get the comment in your, the CEOs, et cetera, might say, well, how do you know your model is, like you said, how is it right? It's not including X, Y, and Z. How do you handle those sorts of things?
Yeah, and so when these models are submitted to FDA, for example, but also being used internally, there's also an examination of the models that were discarded, or the pieces that were discarded, or the hypotheses that were explored. And so all we can say at that point is that this is the model that best represents what we have, the data and understanding of the disease. And that these other hypotheses that were tested were not supported as well with the qualitative and quantitative information we have about the disease. We can't say more than that. And we have to list the hypotheses we tested so that people will know, we didn't test everything, and they might have an idea for something that was not thought of. And so there's a caveat. So it's all about context of use. It's all about saying what we can and cannot. It's that part of that uncertainty quantification. It's talking about, because we focus on this disease, the parameter estimation and uncertainty. Model uncertainty probably is much bigger. And so we want to also give that list. And this is also well established in the statistical modeling space, where we're just putting curves through data. And so we say which functions we tested. What kind, did we use a Michaelis-Menten, or did we not use, did we use a nonlinear clearance, or did we just test a linear clearance? But you have to show what you've considered so that people will know whether or not that their hypothesis was ruled out, or their hypothesis was not even considered.
Got it. Thank you.
Great point. Yeah, thanks. And I think I've pulled the chat, and I think there might be a few questions in there too. Yeah, global sensitivity analysis, very expensive for an academic researcher. But so what we do is we use local sensitivity analysis as a first step. So if you've got a model with 1,000 parameters, then yes, do a local, do a one at a time. Do one of these OAT sensitivity analyses. And then what you can do is, if you find parameters that do not appear to be influential in the ranges that we tested, you can freeze them and see if you get the same outcome when you don't freeze. And so you do kind of some preliminary work. And then after that, you do the global sensitivity analysis on the ones that you can focus on, now that you've ruled out a bunch of them.
I wanted to second your comment about global sensitivity analysis. And when we do these multi-scale simulations, which we do, which parameters are most sensitive changes it completely depending on which region of parameter space you're in.
Yes.
If you're in the region of excessive cell proliferation, then the drug target will look like one thing. If you're in a region of normal behavior, the drug target to maintain things would look totally different, because the sensitivity, the local sensitivity to behavior is so different in different regions. I'm sure Gary's nodding his head that we all run into this. And so I'm not trying to I absolutely endorse your view of global sensitivity analysis. I don't always know how to do it, because the simulations are the simulations we run are not OD ones, but they take days of computer time per simulation.
Yeah.
So millions of simulations is expensive.
Right. So I would argue that by adding a control, you're changing the model structure by definition, right? But you're adding a feature to that model that otherwise wasn't represented in a particular way. And therefore, what the assumptions you had made about what the parameter space or sensitivity of that system prior to that particular intervention cannot be, or not conserved, particularly given the nonlinearities of the system.
That's a good point. Something I haven't done much work in, but I think you're right about that. And back to the point about the expensiveness and the local sensitivity analysis. Yeah, so we like to say that the rank order in your sensitivity, your global sensitivity analysis, or global sensitivity analysis, it is a function of the parameter distribution, not just the range. Because we do not usually work in a space where parameter distributions are uniform. Our space, that in the QSP model world, it's almost always log normal. I have seen only one time where maybe there was a credible example that wasn't close to log normal. But otherwise, our parameters are log normal. And so the sensitivity will depend on the distribution that's chosen. So it's a function of that. But yeah, okay. So then back to the comment about the so the comment about the control is talking about that left and right hand side. You do the sensitivity analysis over here, but then you put some therapies, interventions in there, and then the sensitivity analysis would be different, right? Yeah, good point. Yeah, maybe we should compute some of that.
Any other questions for Helen?
So at a practical level, I have a model. The model is used to help to design the clinical trial, phase 1, and we begin administering a pharmacological agent, and a bunch of side effects, or one side effect, shows up very often that the model didn't predict. So what happens to the model? Do you say, wait a minute, this mechanistic model doesn't model all of the mechanisms or something, because we're getting unexpected results. And it kind of relates to, at the top of your list of a model's usability, the validity word keeps showing up. What kind of validity are you trying to get? If it's predictive validity, then we would say, well, wait a minute, the model did not predict what we observed in a small sample. So how do you balance that? And what do you mean by validity?
Yeah, and so validity is qualified validity. So that's why we make a plan. We are very careful to state the assumptions and what was tested, what wasn't, and then to state what we can with the level of confidence that we can. And so it's a really good point that we do have this happen all the time. The model makes a prediction. The results are not quite what we predicted. So there's an unexpected side effect. Now we're getting better and better at that, I have to say, especially the side effect side. There's a company in North Carolina that spun out of academia. It's called DILIsym, Drug-Induced Liver Injury Simulator. And they have gotten very, very good because they know mechanisms to such an exquisite level of detail, transporters and inhibitions and competing transporters. So if there's a new drug, if they know the molecular structure, they can sometimes make a prediction, or I should say it's not the molecular, it's from the in vitro and in vivo data. They will make a prediction about human because they know the differences in expression levels of receptors, transporters, all of that, those differences. And they will make a prediction before the study begins. Of course, you have things that are not expected. And so that's what we call the learn, confirm paradigm. Lou Shiner, I think, coined that term in our field. Learn, confirm for modeling, where you make a model, you make a prediction, you test the prediction. If it's wrong, you go back and re-examine your model. But these models are typically, they span the course of the development space. So they'll start in discovery with very few parameters, maybe a minimal model. And if that compound moves forward, then the model will move forward too and get more support. More data has now been used to go into the model, and then more mechanisms will be added and more detail and more confidence as we go forward in through the development. So, so there's a lot of confidence in the models at the late stages. There's more confidence because it's been tested in various settings, and we have some understanding of the different animal models or in vitro systems, the microphysiological systems that can support these models. And there was recently, actually, FDA just approved last year a therapy based solely on a mechanistic model. Olipidase alpha was approved, and it was approved for adults, but it had never been approved for pediatrics. And there were some questions, and they used models, mechanistic models, to test a handful of different hypotheses or the reasons for the differences they would see in the two populations. And what they came up with the data supported, with the model, the data supported that the stage of the disease was simply earlier in the pediatric population. And so they gave the approval for the dosing based on that. It was a rare population, rare disease. And so it was really going to be hard to do those studies. Now, I will say, in defense of these mechanistic models, if we don't use these mechanistic models, what do we use? And in the past, we simply jumped. We took a leap of faith. And so the mechanistic models are intended to guide those jumps, those big leaps from one system to another, in vitro to human or others. So, and it's a way of, in a rational form, we put all the information we know, qualitative, quantitative, in vitro, in vivo, first phase of pilot study in humans. And we build it and get better as we go. So I would say it's one of the ways to support drug development. It's not the substitute for drug development, but appreciate that point you brought up.